All right, everyone. We are going to get started with day 2. Thank you all for those of you who were here yesterday and for the awesome first day. And today, I think, is going to be just as great. Starting us off is going to be Denise Kirschner telling us about her work on multi-scale modeling. Thanks, Denise.
Okay, good morning, everyone. Can you hear me okay? Okay, great. So I'm going to talk to you about something a little bit different, and that is using something called multi-scale modeling to also look at spatial aspects of disease. And so you can sort of think of this, the title of this talk is, can we figure out what gets you to the images that you see, what's in the images, and what happens after the images were taken? Okay, so it's a dynamic thing. So what is multi-scale modeling? A multi-scale modeling, you can see I have an image here of something going from the molecular scale all the way up to the population. So the idea is that you build models at individual scales and then you thread them. And they usually represent different physiological compartments, or they can even go much south in molecules and do atomic level modeling, but there's just an idea, at least of the things we focus on for disease. Of course, the reason that we need to do things multi-scale, because if you just study something at one particular scale, you're not going to get all the information because, it doesn't exist in a vacuum, right? It's connected north and south and east and west to different aspects of the body. And importantly, this is necessarily mechanistic and not AI, okay? So even though AI is really important and we can post-process data or synthetic data that we generate with AI, it is not a machine learning tool. This is actually mechanistic modeling. And so I'm going to do this example, and the work that we do focuses on tuberculosis. So I'm just going to spend 1 second telling you about TB. TB is the #1 cause of death in the world. Yes, even beyond COVID, more people have died of TB since the beginning of time than any other disease. And there's still about 2 million deaths a year, which is the same number that died just in 2 years from COVID. But the amount of money and attention, of course, that TB gets compared to the rest of the world is, compared to COVID is very different. And there's lots of reasons for that. The idea is that there's 3 different disease trajectories: either clear infection, you have primary active disease where you're coughing up your lungs, like Doc Holliday, sort of a thing, or you have latent TB where you have it for your whole life and you may have a chance of reactivation later in life. There's a 10% chance lifetime reactivation, but if you have immunosuppression or HIV, it's a 10% per year reactivation. So we have this fabulous colony of 300 monkeys that are housed at University of Pittsburgh, that mostly Bill Gates and NIH pay for. And we collaborate heavily with the Flynn and Matilla labs on this work. And we have this beautiful animal model that recapitulates the disease. Mice do not recapitulate it. So we have this very expensive animal model. Though there are many mice studies done, it's not the same etiology in that you see in pathology as in TB. So the nice thing about what we get with our collaborators is 2 different types of spatial data sets. The first one you've heard a lot about on the right here is, immunohistochemistry stained slides. And those are wonderful. And we can tell Josh or Joanne exactly which 3 or 4 colors that we want, and they stain them for us, and we can, get images that look like that. When I tell you a characteristic of most of these granulomas is this dead necrotic core, which we sometimes refer to as caseous necrosis because it looks cheesy. Caseum is Latin for cheese. But we also get PET-CT scans. So you give a glucose that is taken up, FDG, and it's in metabolically active cells, it sort of lights up. We don't know yet if it's a marker of bad or good. Nobody understands actually what the FDG actually is a marker of, but we can at least identify all of the different granulomas. And the big hot thing in the center is a lymph node, is a cluster of lung draining lymph nodes there. The nice thing about that is that we can get a few serial snapshots of that, where of course you don't get anything serial when you get a granuloma. You get, a necropsy and you're done. One other piece of important information is that primates have more than 1 granuloma. And for every bacteria that you inhale, you get a unique granuloma that forms. And so you see here, we barcoded 23 bacteria, instilled them into the lungs, and we got 23 unique granulomas. And the multicolor is a lymph node because, of course, the draining lymph nodes accept antigen from all the different granulomas. And what you see between 4 weeks post-infection and 5 weeks is the splitting of one granuloma, which we refer to as a dissemination event. So, and again, that's not well understood how that granuloma disseminates. These granulomas that I'm telling you about have lots of different flavors, and basically, we don't even understand the reason that they form in different ways. So you can have one that's solid and full of this dead tissue. You can have ones that are full of neutrophils, ones that are just loaded with cells with not much necrotic, and then you can get most of the time fibrotic or calcified granulomas sort of towards the end of disease. But also, if you treat, you get fibrotic calcification immediately. So our goal is to build a multi-scale computational model that captures granuloma formation and function. We've been working on this for about 20 years, and we continuously curate this. So let me just give you a little primer for how you build an agent-based model. Agent-based models require 4 things. You have to have an environment that you're trying to simulate on a grid. So here it's lung tissue, 6 millimeter by 6 millimeter of lung tissue. You have agents. That's why it's agent-based modeling. And here our agents are things like immune cells and bacteria that we're tracking. We know what every individual guy is doing, okay? And then you have also some continuous agents, things that are diffusing, like molecules, like cytokines, chemokines. And then there's the rules for the interaction of all of these things, and there's the time steps that they occur. So here in our image, you see that we have our lung tissue, we have our cells on there, our bacteria on there, we have things diffusing on there, and the micro compartment of our simulation is about the size of a dendritic cell or a macrophage, about 20 microns. And so that means 2 T cells could fit in one, or a single large cell. And so then there's a lot of rules, thousands of rules. And if you know anything about our collaborator, Joanne Flynn, she keeps us very much truth grounded in the biology here. So we update these rules regularly about how do cells move on this grid, what are they secreting, which cells should be involved and not involved. So things have been added over the years into this system. But here's the amazing thing about an agent-based model, which is used or object-oriented programming, okay, is that you put in all these rules at the cellular scale. You say, oh, when a T cell meets a macrophage, it gets activated. All of these things are in there. It leads to the emergent behavior of a granuloma. We do not tell it, draw a granuloma. We just say, let these guys interact, what happens? And so when we do that, here's our immunohistochemistry. This is our simulation of a granuloma. And what is great about this system is, of course, we don't just get this static. We actually get, well, where's the movie? There it is. Okay. We don't just get a static image, but you see we have a lymphocytic cuff of those small little pink ones. We have the macrophages. We have macrophages that are activated in blue, ones that are infected in orange, ones that are really badly chronically infected in red. We have caseum developing in the center. There's bacteria everywhere in there. And so we are able to generate this image without telling it to do that. It's an emergent behavior, a property of a agent-based model. And so not only do we match to what kinds of data we see from the non-human primates, we can do that also match temporally, from 600, granulomas over time. We can get bacterial dynamics, T cell dynamics, macrophage dynamics, and make sure that these things make sense in both space and time. And so I'll show you a simulation here. I don't know how to, where's the mouse? Oh, here it is. So you can see that if we just drop in one bacteria into the center of our grid, because it looks prettier when it's in the center, but doesn't have to be there, we can see this granuloma. Now, the first time I showed this at a Keystone meeting about 15 years ago, people that study TB are like, oh my God, because you see lungs are black boxes. These are not things that we can study regularly. So how do we get information about what's going on in there? And if we can understand the processes and the mechanisms that leads up to the development of this granuloma, maybe we can also understand how to, help control the disease. Okay, so just to give you an idea, I showed you 4 very different types of granuloma that were taken from the monkey. Here's 4 different types of granulomas that we simulated. So here's one that contained, it's beautiful, nice solid cellular. There's one that's disseminating. Here's one that's cleared but left behind a huge inflammatory signature. And here's one that there's not much going on, but you have tons of bugs because the macrophages are not being activated. Even more so, we can actually look at gradients of molecules that are underlying that. So there you see a TNF, that the TNF gradient underneath every one of those granulomas and what it looks like. So we're able to glean all this information for any cell at any particular time, which is, a very useful thing. One other thing we're able to do actually is also look at the PET-CT and try to understand better how the signature starts. And you can see that for a single granuloma, we're putting it, Josh Mantella, our collaborator, is trying to better understand what drives the PET-CT color in these monkeys and images. And so we're working with him to do that as well. All right, so now, if you believe that we have this model that works and it generates granulomas, the question is, we should be able to ask with mathematical tools, what drives these different granuloma behaviors? And to do that, our analysis tool is something called uncertainty and sensitivity analysis. So uncertainty says, how bad is your model if the parameters that you're putting in there are really wrong? And sensitivity says, which parameters are those? Okay, so it's a very powerful tool, and anybody who's a mathematician in the audience, I'll just tell you that it really almost identifies transcritical bifurcation and hop bifurcation parameters, really can get in there and find this stuff, even though this is not a mathematical system, it's a computational model. But the cool thing is we can do this over time. So let me tell you the result of that. So suppose we want to know what's driving CFU inside these granulomas, that's bacterial load. So here you see over time, these are the parameters that ended up being significant in this nonlinear correlative system that we use, because these are highly nonlinear models. So we have to use something known as a partial rank correlation. And so you see in the first 50 days, there's very different behavior of which parameters are driving the development of that granuloma, as opposed to later, once the granuloma is established, who's keeping things in check. So that's the first piece of important information. The second thing is, in those early days, you see somebody, I don't know which one is green there, one of the parameters starts very high, the source of TNF, and then actually changes and becomes negative, negatively impacting that CFU over time. And so similarly, there's lots of different aspects there. So we grouped those results into 5 categories, and it's basically, we decided to start drilling down and say, okay, TNF looks really important. It's showing up in lots of different ways, so let's study that further. So we built models that understand the dynamics of TNF more importantly, and figured out exactly which mechanism was driving it. But the cool thing is that this was already known in mice, monkeys, and in humans who take anti-TNF drugs like infliximab or etanercept. These people suffer reactive disease. So we already know TNF's important. This is just another way to understand that the model does predict that things that are very important in the actual system. Okay, so since we're at a talk about a conference about imaging, I wanted to just share with you about 3 ways that we use MIBI-TOF data and collaborate with Mike Angelo and his group. Geographical information systems, we work with Russ Butler and his group. He's going to be talking this morning. And then also somebody mentioned MALDI imaging yesterday, so we also use some MALDI imaging as well. So the question is, how can grandson predictions help advance data from other spatial methods and vice versa? So you saw this panel yesterday. Aaron sent us this, and this is typically the way we get information from the MIBI group. We kind of think of it as this, is that we, instead of breaking it up, this way by every individual thing, we kind of group them in 4 categories. What are things related to cell identity? What are things related to microenvironment, to signaling, and to regulation? And that corresponds in our story to things that are agents, things that are on the grid, and things that are within the rules in our model, okay? And so you can see that there's a lot of overlap between things that are in the panel and things that are in our panel as well, all right? But there's also some important differences between what's in the MIBI panel and what's in the GrandSim panel. So in the top there, I'm showing you things that are in the MIBI panel but are not currently in the GrandSim panel. And then similarly, these are all things that are in GrandSim that are not currently able to be identified in in the MIBI panel. So we really think that if we look at the associated features, and Candace, there's your figure. The associated features, that are coming out of MIBI and the associated features that are coming out of GrandSim, if we can somehow put these together with the metadata on the monkeys, we're going to be able to say a lot more about this system. And maybe we'll have to add more analytes into GrandSim, or maybe there'll be additional things that they look at in the panel so we can really start to understand what the mechanisms are that are driving the different types of granulomas that we see. Importantly, which ones control versus which ones don't control infection. So the second story is how do we use this micro-GIS? And I know he hasn't explained this yet, but I think it'll be obvious from the picture. So in TB, one of the interesting findings that Joanne has seen in the granulomas is that there's very low numbers of MTB-specific T cells active in the granuloma. Okay, very low numbers. Like 10% of the T cells that are in the granuloma are responding to any kind of stimulation. So why is that? It could be because they're exhausted. Everybody thinks exhaustion. It's not. We've done a study with Joanne and modeling. It's not exhaustion. Maybe you don't have enough specific T cells. It is not that the T cells that are there are not specific. So what is going on? So we built a model with GrandSim that said that it's because during the peak of activation only, and this is at day 40, but this is over time, that the average distance between T cells and macrophages is too great. Only about 30% of T cells are able to get near a macrophage. And later on, of course, those numbers drop over time as this granuloma grows. Now, if you know anything about immunology, T cells and macrophages have to interact to get that activation event to happen. And so that is actually what's happening there. And if you've looked at granulomas before, there's this characteristic lymphocytic cuff around the outside of it. And why is that? Probably because those T cells cannot get in and penetrate inside this solid structure. Okay. So this was our hypothesis. So we asked Russ and his team to look at 4 different granulomas. So you see them in this column here, the IHC, and he was able to, and he'll tell you later, identify what were the T cells, what were the macrophages, and where they were overlapping in these granulomas. And what we showed is only about 8% of the T cells were touching anywhere near a macrophage to be able to be stimulated. So there's your answer. That's why the levels of activated T cells within a granuloma are so low. Now with modeling, we can do really cool things like, adjust things a tiny bit or let T cells squeeze a little bit better. And if you could get T cells near macrophages, your granulomas would sterilize every time. So clearly that's a main issue in a success of a granuloma. Okay, so the last piece I want to tell you is about the MALDI. And so one thing that we use our GrandSim simulations for is drug treatment. And the cool thing is we can use the same granuloma, treat it with lots of different things, but we're using the same exact granuloma every time. So we can really understand the differences. So here's an example of treating a granuloma with rifampicin and isoniazid. And you can see that the half-life of INH, that one's already gone, but after the first treatment, there's still some risk, but notice most of the drug is outside, right? It's not penetrating into the granuloma. That's a characteristic of drug treatments of tumors and also granulomas. And I'll tell you that most of the things I said today about granulomas are probably hold true for tumors as well. So we calibrate and validate all the time, though, to MALDI data. So we get these beautiful MALDI images from Veronique Dartois, where they take these granulomas and do a MALDI image, outline the caseum, and you can see in our synthetic data, we're able to do something very similar and understand why the penetration within those granulomas is different. And we can also match to temporal data that's generated in that as well. Okay, so I'm just going to switch gears for the last 2 minutes and just tell you that I've been talking a lot about granuloma, but as I told you from the beginning, there are multiple granulomas in every host. In fact, there's about 13 on average in primates. And then, of course, we're interested in what's going on with the population. So our recent goal has been to create a virtual patient and also to create a virtual cohort where we can do lots of things like vaccines and drug studies, because there's a big push with the Gates Foundation and the NIH and the World Health Organization to improve treatment for TB, and so we're part of those studies. So creating what's sometimes now called a digital twin or a virtual patient can help us advance many aspects of these things. So what we've done is we took, we got the people at Pitt for us to take a healthy non-human primate and digitize it and give us the XYZ coordinates so we can recapitulate that in our computer system. So this is a primate where we have the lung, a healthy lung, and the torso of a primate. I should tell you that for pulmonary TB, which is what most of the world has, so a fourth of the world is infected with TB. Basically, 90% of them have pulmonary TB. If you have extrapulmonary TB, things are going really bad, or you're most likely a child. So this is pulmonary TB is taking place in this compartment that you're looking at: the lungs and the draining lymph nodes and the blood that feeds the pulmonary area. So what we are able to do now is not only track single granulomas, but track the development of multiple granulomas over time, and look at how the cells are moving between the lymph nodes and the blood, and understand better what's going on in the entire host. And we can also look at these things over time. So here I'm able to show you the CFU for, in 500 hosts, what's the CFU in all those granulomas, what's the CFU per host, and what's the total number of hosts doing over time, and who's sterilizing and who's doing what. So we get basically a multi-scale view from granuloma and molecules and cells and tissues all the way up to population. Okay, so in summary, basically what I've told you is that multi-scale computational modeling in spatial biology is an additional tool for spatial analysis. Models are built at various scales and are appropriate for the use at whatever scale you're interested in, can be combined with other approaches for synergizing and maximizing analyses to identify mechanisms. And that's what we're looking for, is mechanism. And static imaging together with this dynamic modeling can tell us more than the sum of the parts. So have an amazing group at Michigan. Park Sheet is here with me today. He's a postdoc, and we have funding with LEAP and Gates Foundation, NIH, NSF, and yeah, fabulous collaborators that provide all the wonderful data that we have. So anyway, I'm happy to take your questions.
That was really awesome, Denise. If anyone has questions, you could please walk up to the microphone in the center. I'll start with one from online. How did you determine the optimal number of primate samples in order to have confidence in your results?
Did you say primate samples?
Yeah.
So yeah, well, that's not really a question like for me, but what I will answer it by saying that we have thousands of simulated granulomas and simulated monkeys that we can do, so we're not limited anymore. The 300, the colony that we have, and 600 granulomas, those are statistics. Those are all they do. What's it called? Analyses where you determine how many you have to. Yeah, power analyses, right? So that's all done. We have a statistician on staff that figures those things out. So yeah, Mike.
Love the talk, of course. So two questions for you. The first is you mentioned that you think this stuff could apply very well to tumor models.
Yes.
So the first question I have is how essential is it for this type of approach to work well to actually have an animal model with a controlled time zero, and where you actually have sort of ground truth on the temporal trajectory of it? Like, for instance, do you think you could use these types of approaches looking at primary resections from human tumors where you don't necessarily have
ground zero?
Right.
Absolutely. I mean, I think a lot of times, we have access to some human data now. If you start in the middle, one thing that modeling can do is say, how did we get to this granuloma? We can project back and we can project forward, but absolutely, just having even sometimes single time points is enough to just kind of give us a ground truth of where, what's going on. And then we can try to build the story around that with the modeling, for sure.
And then second question is I think agent-based modeling is like this, blue sky approach that really hasn't been tapped enough on. I hardly see anything about it on the cancer side. And so for people that want to look at these approaches and learn more about them, what kind of advice do you have for, is this like a huge undertaking? I mean, it sounds like you guys have been working on this for a really long time, or
no. So let's see, is this connected to the internet? because I can show you something really cool if it is. I don't know. It's just got this fancy Stanford medicine thing, so I don't know how to get out of that. But because I'm not a PC person, maybe. But so there are amazing toy programs out there. The one everybody should just look up is called NetLogo. So it's just netlogo.com or something like that. Just go to it now, and there are like 50 built-in agent-based models that you can watch people doing game theory and all different kinds of things there. And you can build your own. You'll say, here's 4 things that I want to look at. And you put it in and you can play with it. You can build an agent-based model in an afternoon if you want to for just about anything. We don't use those forms because ours have gotten so complicated that it can't do a lot of the really heavy lifting that we need. Our models are ginormous. We have 100,000 lines of code in our C++ object-oriented code, but the NetLogo, we love NetLogo. And when I teach courses on this, that's what we use with the students. So I think you'll have fun playing around with that. And there are people, so the lab of Alexander Popel at Johns Hopkins, graduate student from my lab, went and took the technology, and Sasha is a cancer modeler, and he took grants and turned it into a tumor model. And then looked at PD1, PDL1 dynamics on that. So that's published. You can look up. Cheng Gong is the first author on that, and Sasha Popel is the last author. So you can see a paper where someone did an agent-based model of a tumor, and there's other people working in that, but that'll be a good bridge point if you want to look at the literature on that. I hope that helps. Yeah.
Thank you. That was fascinating. Just wanted to ask, the lung microbiome plays a role in everything
Sure does.
more and more now. Are you incorporating any of that, and how can you get that in part of your system there?
Yeah. So there's 2 things that we're very interested in right now: lung microbiome and tissue resident memory cells, because they're probably even playing more of a role, especially if there's been a vaccination event, right? So right now, Joanna's actually trying to map the lung microbiome in the primates so that we can then understand their role better in what's going on. So those are the 2, that's our sort of frontier right now.
Okay.
Thank you very much.
Hi, really great talk. I know you didn't get to talk about this too much, but I was curious. So for the virtual patients that you guys are working
Yes.
on creating, how are you ensuring that like the patient is going to be more like representative of like who is primarily impacted by TB? So including like gender, racial diversity, and things like that, ensuring, since we know a lot of like deep learning and AI systems are still very biased and not inclusive.
Yeah, no, that's a really good question. The cool thing is that, in the modeling, we can control every aspect of it. So we did a model, for example, during COVID, where we looked at, the dynamics at the population. It was a population model, network model, and we had, lots of diversity in there, in age and behaviors and race, all of that. And we took it directly we did one city. So we downloaded all the demographic and just threw that all into the model. So it was about as accurate as you can get. So we care very much about those details, and it makes a huge difference in diseases like TB, where, if you're in Africa or India with TB, or you're in America with that disease, not only is your availability of treatment and all those things, but your background, baseline is going to be very different based on nutrition and health and access to all of those things. So yeah, so thank you. It's an important question, but that's all doable with the modeling.
So you had a slide when you were kind of simulating drugs. So I was wondering if with your model, are you able to kind of like maybe new drugs that aren't that haven't went through clinical trials, can you kind of put those drugs in there and see how they would do? Or maybe try to create new drugs that maybe you can get the parameters that would be needed to treat TB? Is that something you could do with your model?
Good question. Yeah. So we've done right now, the Gates Foundation is paying us to do a study where we have 20 TB drugs that are possible drugs. They use these same 4 all the time right now. When you show up in the clinic with TB, you get these 4 drugs, but there's an arsenal of about 20. And so, but if you think about the number of possible combinations and permutations of 20 drugs in groups of 4, 3, 2, 1, whatever, it's like 10 to the 17th. It's crazy how high that number is. So we're using the modeling to kind of whip through all of those, possible regimens to predict, oh, you guys should be looking at this set of regimens. These are the promising regimens. Now, about different drugs. So with the sensitivity analysis, we can look at a drug like INH and say, okay, if this drug were to just improve its C50 just a little bit better, or if you were able to make the absorption just a little bit slower, then that would change it. So then we can pass that information to people who are pharmacologists, right? And say, if you can just, there's a new drug like rifampicin, which is a little bit different than rifampicin. One of the other drugs I talked about today, rifampicin. And so, but because they've just changed part of the structure of what that drug is. And so we can make suggestions to how improvements to the actual pharmacology of the drugs as well with the modeling. So we've done that as well. Yeah.
Cool. Thank you.
Thank you. Great questions. Okay. We done?
All right. Thank you, Denise.
Thank you.
In the interest of time, we're going to move on. Let's give her a round of applause. And next up, we have Nils Gehlenborg from Harvard Medical School, who's going to be telling us about the awesome work his group does creating Vitessce.
Gonna try this. Can you hear me okay? Everywhere? Awesome. All right. Sorry, was not good. All right, good to go. Fantastic. So 20 minutes. I don't have to do the math here. Fantastic. All right, I'm gonna tell you a little bit about our data visualization work. And before I do this, I just want to briefly highlight a little bit how our work is maybe a little bit different from what many of you are doing and interested in. So the focus of my research group, which is called the HiDive Lab, and HiDive stands for Humans in Data Integration, Visualization, and Exploration. So the focus is the human, the role of the human individual in data analysis, visualization, et cetera. So the questions that we're trying to answer is, how do we get data into the human so that the human can take some action? And we do this, unsurprisingly, right, through the eye into the brain, where we are hopefully able to make decisions and take actions. And so we have been thinking about data visualization problems across the whole spectrum of biomedical data over the years, starting with molecular data, genomics, transcriptomics, et cetera, where we've developed a couple of tools. Some of you might be familiar with a few of them that are out there. So I just want to maybe highlight HiGlass, which is originally started out as a tool to visualize genome interaction matrices, but now it's sort of become a full-blown genome browser that is fully web-based and highly interactive. And Gosling is a newer project that is essentially a grammar similar to the grammar of graphics, which some of you might be familiar with, and particularly if you ever used the ggplot package in R. So Gosling is a grammar to use sort of the same approach, but to create interactive genomics visualizations in very diverse ways, including what we have down here in this new tool called Chromoscope, which is a structural variant viewer. I know nothing here has to do anything with spatial data, which I'm getting to now. In terms of single cell and spatial data, we've developed Vitessce, and that we'll talk about, but there are a couple of other tools in that context, Avivator and Vizar, that I'll also mention briefly, that are more lightweight components that you can use very easily in your own software development efforts, in case that's something you're interested in doing. We've worked on sort of patient-generated data as well as EHR data, created tools, not for other scientists, but for patients to help them interpret their own data and use those visualizations sort of as communication tools for a conversation between a patient and provider. We've thought about questions around how do we visualize cohorts I sort of started in my postdoc thinking about TCGA, which still is a very rich source of data for many of us, to essentially create tools to discover subsets of patients that share common molecular or clinical patterns in their data. Some of you might have heard about upset plots, which is another project that we've been working on. And during COVID, we also got into sort of visualizing data for whole healthcare systems, basically. And so, yeah, going from the small to the very, very, very large is, the questions always center around how do we make this information accessible to human. But let's talk about spatial data and spatial biology. So thinking about single cells all the way to organs, and we kind of take an approach, and sort of our work is really informed by our experience being part of many different consortia that try to build these cell atlases. So we're part of the Kidney Precision Medicine Project, the Human Tumor Atlas Network, the Cellular Senescence Network, HubMap, as well as HCA. And as you know, these Atlas efforts are somewhat different in terms of what they focus on. So some of them are more focused on a disease or a specific organ. Others are looking more at particular mechanisms across many organs. HubMap is sort of trying to build this normal reference, and HCA is sort of doing all of the above. And so after having worked in these projects for some time, we've tried to sort of distill what are the key questions when it comes to visualization of spatial biology data. And so, you know, the goal always is to characterize and understand tissues at sort of single cell resolution. Oops, these slides are in here, even though they shouldn't be in here, but I'll go through this real quickly. So we have tissue-specific cell types and their functions as a question, right? We want to identify the cell types, and we want to understand what these different cell types are doing in tissues. We want to understand diseases or changes in cells. Yeah, we want to understand which regulatory networks are involved, cell-cell interactions, and all that. And we're also interested in sort of, you know, what is common across tissues, and how can single-cell and spatial single-cell data help us with that. And so the challenges for visualization that we identified are, first of all, in this space right now, everyone is sort of doing their own thing. And although there are efforts to standardize things, we have to be able to support many different data modalities and support many different biological questions, right? I think, Mike, was it you yesterday? Is that you have to tell your collaborators you don't need to do spatial necessarily, but it seems like everyone is trying to answer their specific question with single cell data, or do something spatial. And so that's yeah, what we're clearly seeing as well. So we need to support a lot of different things. We clearly are have a need to support multimodal data, right? So I think just looking at an image is not sufficient. We have derived information that we need to be able to visualize as well. And so in our work, we handle this by essentially providing setups for multiple linked views, and I'll show you what that means in a moment. We also, and this is really important from my perspective as someone who thinks a lot about building tools for other people to use, we need to support different kind of analytical environments where our visualization tools could be embedded and could be part of sort of a bigger workflow. We also care about our end users being able to deploy their own visualizations and share them with collaborators, et cetera. So we need to think about that too. And finally, data formats are a big thing. We have, thanks to the organizers, a great talk coming up, I think, later about spatial data, which is an effort to standardize things a little bit more. But nonetheless, there are a lot of different data formats out there that need to be supported by visualization tools. And so what I want to do is I want to tell you about Vitessce, as you've probably gathered from the title, which is our visual integration tool for exploration of spatial single cell experiments. That is an acronym, actually. In the next couple of minutes, I will try to give you an overview of what Vitessce can do and how you can potentially use it yourself in various different settings. I do want to highlight some of the team members. Wow, really huge here. Mark Keller, who's a PhD student in my lab, and well-deserved to have a big picture like that, because he's really the one who's driving Vitessce. This is his main project. Trevor Mance and Elon Gold both contributed significantly to the image visualization aspects in Vitessce, which preceded sort of the Vitessce project by a little bit. So the tools. So there is a whole ecosystem of tools that goes into what Vitessce actually is and how Vitessce can be integrated. So as I said, it started actually with image visualization. So we developed this library called VIV, which nowadays really stands for Vitessce Image Viewer. And we built this library because we realized it's really hard to take high resolution multiplexed images and visualize them in a web browser, right? Where you need to be able to change the channels, you need to change the color mappings, et cetera, on the fly. There was just not a very elegant solution out there that was lightweight and easy to integrate into other tools. And so we built this thing. And then Avivator is sort of like a demo of Viv. You can play around with this, but it's also a very useful little tool if you have a OMI TIFF image or OMI NGFF image laying around somewhere on the web. You just point this tool to it and you can visualize it without doing anything. That's what it looks like. And then Vizar is a sort of a really lightweight plugin focused particularly on ZAR-based image data that you can embed in, for example, in this case, to Jupyter notebooks and start looking at your data in that setting. Okay, and now on to Vitessce. So Vitessce can take many, many different forms. Many of the Vitessce examples that you'll see will have some sort of an image in that, like, so there's some spatial component, but it doesn't have to, right? You can also just use it like the viewers that are available, for example, in Cell by Gene, right? So it can just do UMaps if you want, and many other things. So here is just a Visium example where we have two linked views, one of them showing sort of the cluster assignment, and the other view just shows the quantification for some expression level of a marker in here, and we have a heat map. And so, you know, that's sort of one example of what Vitessce could look like. And then what I want to emphasize, Vitessce can be embedded in many different environments. So here is an example from the HubMap data portal, just a screenshot that I took this morning. This is a codex data set that you can find in the portal. And here it's directly embedded. And so whenever you look at a data set in the HubMap portal and you're lucky, there's a visualization with Vitessce integrated right there. It's also possible to just build your own little web application and embed Vitessce. So we've done this, for example, on the Vitessce website itself. There are lots of examples, and you can play around with Vitessce right in there. But in order to make this tool, which I think is useful for end users in any case, because it allows you to look at your data and interact with it. For end users, that's fine, but that's not the only audience that we're thinking about. We're also thinking about other tool builders, data analysts. And so we have this Python package for Vitessce that allows you to construct Vitessce visualizations on the fly in your Python code. Here's an example in a Jupyter notebook. Here's another example of a different kind of view that you can embed. So genomic profiles is an option if you want to look at those and hook them up, for example, to a UMAP and some cluster assignments. So you can do this with the Python package, and some of you might have had the pleasure of working with this in the context of HubMap more recently, but it's something that we're continuing to develop and try to make more user-friendly and more useful as well. And so because not everyone is using Python, of course, you also support R. So if you want to create an interactive visualization of your single cell data, whether it's spatial or not, you can do this in R code, and for example, in RStudio, you can use the Vitessce HTML widget to visualize your data basically in situ in your analysis environment. Okay, so moving on from there, I want to highlight a few more general points in Vitessce, what kind of data you can bring in. So it is designed around multimodal and spatial single cell experiments, and you can not only bring in cells mapped to, or some sort of quantification of a marker mapped to cells, you can do this for beads or spots or any other type of entity that you're interested in having in your visualization, right? So you could, for example, if you have a segmentation of some functional tissue unit like glomeruli and kidney, you have some metric associated with that, you could bring that into Vitessce as well. And then you can look at glomeruli, like you could look at cells, et cetera. And so we have, and I'm not going to go into much detail here. So we have different ways of configuring the data in Vitessce. So there can be multiple matrices. You can have matrices for, say, cell to gene, and you can have matrices in the same data set or in the same visualization from cell to protein. That's all possible, and you can connect these things in different ways. So this is also where things get a little complicated, but also this is the piece that makes Vitessce really powerful, is the ability to configure these visualizations so that you can link together different views. So for example, you could have two images or the same image twice, one of them serving as an overview and the other one as a detail view. So you would hook them up together so that they move together, but maybe you can control the zoom level separately. And all of that is handled through so-called coordinations, which are not highlighted here. It's one of the little things in this graphic, but these coordinations allow us to create basically any type of visualization that you can think of in tying together different visualizations, heat maps, images, genome browsers, embedding views, and all that. And so that's sort of the secret sauce. in Vitessce, and something that's maybe a little harder. I mean, as an end user, you don't see that, of course, it just works. But for someone who's trying to set up their own instance of Vitessce, this is something that might require some time to wrap your head around. And then, of course, as I said earlier, data formats are critical. We support ANData, MuData, OMI, and GFF. Multivec is a genomics format. And all of these formats are used in the concept of Vitessce in form of ZAR stores. So we need to convert these into ZAR stores, which is not really hard to do, but that allows us to just put data on the web and pull out small subsets if the ZAR store is organized appropriately. And then, of course, there are lots of different view types up here. So scatter plots, spatial imaging, heat map, et cetera. I've already mentioned many of them that you can embed in your visualization. Yeah, and then ultimately what you get is something that looks something like that, depending on what your needs are. All right, and so I just want to show you a few examples. One of the problems in HubMap was that we have a lot of multimodal data sets. We have spatially resolved single cell data, and we have lots of different end users. And we went ahead and basically deployed Vitessce in the HubMap data portal. And so all these examples up here are straight from the HubMap data portal. There's a codex data set, right? So we have the spatial view. You can overlay quantifications. In this case, it's the codex markers in pinkish in that color map. But you can also see the underlying images with the different channels. You can choose which channel you want to look at. You have a heat map for the different clusters down here as well. Apart from that, we can also do fun things, and this is really more fun right now than super useful, but I think it demonstrates the flexibility. So here's some imaging mass cytometry data. It's helpful to see, what the data looks like, but we don't have any segmentations. We don't have any annotations for these data sets right now. So if we had those, we could bring them in, but since we don't have them, this is mostly a pretty picture. Right now, although I think if you were the lab, or if you were from the lab that generated data, you could probably tell a little bit more about this. I can't, because I don't have the information about what was really done here. Yeah, other things that are maybe more useful is this one, and here is an example. This is a SlideSeq data set. So you have both the spatial view in the center, and on the left you have an embedding view. And then on the right, you can see we actually have cell type annotations for these data sets. And you can overlay those on top of these visualizations, or you can show the expression levels of a marker, in this case, UMOT, in this kidney tissue, and you can see that works as well. And there are many other things we can do. I mean, we can, as I said, we can do some standard, visualizations of single cell data on the top right. We can do histology images, or we can also do some light sheet data visualization down here. Again, since those were never segmented or annotated, these 3D views are not super useful apart from pretty. Okay, what I want to emphasize here, so this is only the beginning. These visualizations I just showed you are single sample visualizations. So yes, you can get some insight from looking at these and interacting with these, in particular if you have annotations and segmentations. But yeah, you can bring in clustering, segmentations, cell type assignments, and all these things, but it's only for a single sample. So you can't really do very much in terms of comparing data sets to each other. And so thinking about visualizing multiple samples is something very active project in the lab. So we're working primarily with members of the Kidney Precision Medicine Consortium to develop approaches around Vitessce to explore multiple samples, right? When we want to ask questions about differences between two conditions or trends over time, or other things. And so that's something we're actively working on. Another thing we're actively working on are atlas visualization, right? So how do we think about visualizing a whole atlas of an organ, or even a whole organism? And so in that case, we need to think more about navigating the space. Of course, nobody will have a complete atlas of a human body for a very long time, but we will have many different parts sampled and analyzed, and then we need to tie this somehow together to help people make sense of it. So that's something we're also actively working on. Another thing I really want to highlight is the integration with data analysis tools. So Vitessce itself doesn't do any data analysis, and that's really for good reasons, because we didn't want to get into the business of telling people how to do data analysis or what packages to use. So we made it easy to, as I pointed out, to create your own visualization from whatever computational environment you're in. And so, as I pointed out before, we can, write a little bit of code, in a Jupyter notebook, for example. And once you execute that code, you get the embedded Vitessce view directly in your notebook. You can modify this. So this is actually something I grabbed directly from the HubMap data portal. Because all the visualizations in HubMap portal are generated with Vitessce, I can also give you the code that was used to generate them. And you can download them in form of a Jupyter notebook, and then you can start modifying it. So in this case, I just added a copy of that one view here. I didn't change any of the configuration, but theoretically you could have one of these puck views that shows you the cell type assignments, another one that shows you the quantification for a marker in there. So, and that is done in this way. And so I want to keep this brief. I just might get a little might go a little too deep, but essentially what you do to bring Vitessce into your environment is you write some code in R or Python or JavaScript, and that code essentially just creates a specification that describes what your Vitessce visualization should look like, right? What views should be there, what data should be shown in which views, and how are they connected to each other. And that's this view configuration, which is expressed in a JSON object. And that's the only time I'm going to say that. And then that configuration is just passed on to Vitessce itself, which can be embedded as a React component in a web application or in a Jupyter notebook or in R. So that's really how it works. One thing I want to emphasize again, there will be a talk later about spatial data, but so we are really focused on ensuring that we support all the different data modalities that SCVerse supports. SCVerse is on an effort to make single cell data analysis a bit more standardized. And so there's data, there's mu data, and now spatial data. And we're pretty close to supporting all of spatial data, but we're already supporting mu data and data in Vitessce. And it's something we want to pursue to really make sure it works well. Vitessce doesn't work only for HubMap or for my lab. There are lots of examples out there by now for other efforts. So the KPMP actually integrated Vitessce into their spatial viewer before we even got involved with KPMP. So they have that in there. Web Atlas is a separate effort to create essentially a standardized analysis pipeline with visualization for spatial single cell data out of the Sanger Institute. So they've used Vitessce to as their visualization component. And there are just a couple of other examples where Vitessce was integrated and is available for end users. We are currently working on some new tools towards sort of really biologically meaningful or maps that can be queried in a biologically meaningful way. So here's an example from kidney tissue. This is also KPMP work, where you can see we have different entities segmented out, and you can highlight them in different colors. You can also bring in information about these entities. So here we have different structures in kidney biopsy segmented tubules. There are different types of glomeruli. There are arteries and so on and so forth. And here, I just selected and highlighted the tubules. And then you can see this down here. I overlaid the area of the tubule on top of the tubule. So you can see when I fade out the image here, what the size or the color represents the size of the tubule. It's just not super exciting because you can, in theory, look at it and see that there are some bigger ones and some smaller ones. But with this visualization, you can see this much more readily, how they're distributed. Where are the small ones, where are the large ones? Because now we have an object and we can change its color, etc. Here's another attribute. This is the average cell thickness overlaid on top of the tubules. Here is the luminal fraction highlighted on top of the tubules. And so you can think of any kind of data that you might have associated with whatever stuff you have in your image, and you can bring it in, overlay it. I could also bring in a scatter plot that shows you that, like, essentially maybe 2 dimensions. Like you maybe want to compare area to luminal fraction or something. And so we could easily hook this up in Vitessce. That's the flexibility that makes this really powerful. All right, but that's clearly not the end of it. So where are we all going to go with this, and what are the questions we have to think about in terms of visualization of spatial biology data? And I said like tomorrow, which given how quickly this field is moving, it might literally be tomorrow. So questions in terms of scale, right? How, like, dealing with millions of cells potentially, or not only potentially, that is already a real question. How do we support this? There are some technology questions, engineering questions, but also some questions really around data visualization, because usually there's no point in showing someone millions of things. They probably can't interpret that. Multi-sample data sets, I already mentioned that. We want to bring in more knowledge into these visualizations that help you interpret what you're seeing in terms of patterns in the data, like pathways, for example, anatomical information from reference atlases, such as the Human Reference Atlas and others. And what I'm particularly excited about, and I think what's something that's going to be a huge challenge for visualization is, of course, 3D volumes of single cell data, right? So we need to have 3D segmentation somehow to be able to remove layers or sets of cells or types of cells that we're not interested in, or bring certain things in. We need to think about, of course, again, the scale. But that is, I think, something that is in the 3D visualization realm going to be challenging in terms of dealing with tens of thousands or even hundreds of thousands of cells in 3D volumes. We will have multiple modalities potentially at some point. And of course, also for each modality, we might have thousands of measurements. If you just think about transcripts, right? There will be, I don't know, potentially 20,000 measurements per cell in your 3D volume. And to bring all of that into one environment that is user-friendly and can be deployed in different environments will not be completely trivial. So if you're interested in working on those problems, have a great lab, a lot of fun together. So we are extremely collaborative. We work with experts who are developing assays and technologies. We work with computational experts. We work with other visualization experts, and yeah, it's a lot of fun. So shoot me an email if you're looking for some next step in your career. Would be happy to chat with you about opportunities that we have. And finally, want to thank everyone, and of course NIH for a fun day. Thanks. Ate up all of the time plus then some.
All right, if there's any questions in person, please walk up to the microphone. I'll start with one from online. There's a couple different flavors of this. Basically, what's the best way to get started using Vitessce?
Yeah, I think the best way to get started is to play around with some of the tutorials that we have put together. I think the Python package is probably the best starting point. That's the most developed part. But if there's someone who knows how to create a web application, that's also reasonable. But I think it's a little bit harder on the JavaScript side to figure out how to hook up all these different things.
Okay, got it. Next question. Are you planning to incorporate segmentation packages directly into your program?
No. So we won't do that because there are just too many of them out there. So what we will do is we'll work with the community to figure out what data formats should we be using. And then we'll make sure that we can load those formats that are most popular, and then talk to the people who develop the packages that they need to provide the outputs in a certain format. I think that's for this project the better route. I should add though, and I left it out of this presentation, Vitessce has been used in a number of projects essentially as a front end to an analytical back end. So we have some examples for cell type annotation where we build a server and a service that we hooked up to Vitessce. So Vitessce felt like an analytical tool because there was sort of no sort of separate step to load your data in. So that's possible in that case if someone really wanted to build a tool for interactive segmentation, although there are already some great ones out there, so I don't think anyone needs to. But you could theoretically set up a service that will do the computation and send the results back to Vitessce and display them in there.
Awesome. And then question sort of related to what you mentioned, and I'll build on it a little bit. This is asking about converting to ZAR, but do you have general opinions on the best ways to format your data that are maybe going to be future-proof? People don't want to be constantly redoing their code so that it's supporting the latest format.
Right.
But there is some community benefit to having some standards. What do you guys feel? Is it converged yet on anything, or
So we're in a way big fans of the work that the SCverse community has done in terms of data, new data, and now spatial data. So I think trying to stick to those sort of standards that were designed by people who know a lot about the analyses that people want to do, and who are leading in the field, I think is very helpful. I think that's what we're doing. We definitely see that, I mean, data formats, even though we care about data visualization primarily, but data formats are such a big challenge in visualization, because if you don't organize your data in a way that is useful for visualization, your visualization tool is not going to work very well, and then you have to still do some conversion. So I think we have opinions, but there are other people out there who have even stronger opinions and who have maybe more expertise in this specific data domain. So we trust that the community will do the right thing and will work with people to make sure that our tools support whatever standards the community develops.
Awesome. Let's give a round of applause for Nils with a
Thank
great
you.
talk. And our final speaker of this session, oh, I forgot to pull up your talk title. Hold on. Is Luca Marconato, who's going to be talking about spatial data, new format for solving all of our problems with interoperability.
Hello, everybody. I'm very happy to be here today to talk about the Spatial Data Framework, a framework for storing, processing, and analyzing multimodal spatial data. And let's start directly with an example to motivate the need for a framework and consider data from Visium 10x Genomics. The data comes from the Space Ranger output. Let's imagine to have it stored on disk in the bottom left. The technology, as you know, comes with HNI stained image and the gene expression data. And if you want to start your analysis, you will probably begin with visualization. You can apply image processing tools on the image part of the data, or you can also work on the gene expression part of the data. When you apply these tools, you are interested in saving the results to disk, which can, of course, be done. But when you do this, you may start introducing some heterogeneity to the storage. Now the data is not the raw data anymore. It has a different format. And if you want to visualize the data, of course, it's possible, but it requires some data manipulations. And if you continue downstream, now maybe you want to apply a tool from a different programming language. What you obtain very quickly is a Frankenstein object that gets really complex to visualize or process. I talk about one sample from the Visium technology, but there are studies. This is a study from Lundenberg et al. With multiple Visium samples, and this gives complexity because you have multiple annotation to attach to the samples and also very large images that are heavy just even to visualize, to load and visualize. Visium, of course, is just one of the technologies available. There are every year multiple new texts developed, and even the formats of previous technologies get changed over time. And the final axis of complexity that is not for all the contexts, but sometimes there are biological systems in which you want to apply multiple spatial technologies to have a multimodal view on the data. So, for instance, this is an example from 10x Genomics of a breast cancer study analyzed with Visium and Xenium, but also with single-cell RNA-seq. And the reason is that different technologies can give orthogonal signals. From Xenium, you can get very accurate cell locations and boundaries, but you have not many genes. Still, you can use single-cell RNA-seq for finding cell types which can be mapped accurately to the Xenium data. Then you can use Visium, for instance, being transcriptome-wide to study cancer clonality, and you can look at the image to annotate anatomical regions that can be used downstream. And this shows the need that we can't just look at the data as individual components, but we want a multimodal representation for visualization and downstream processing. The challenges that I described motivated us to join forces. And with us, I mean the SCVerse team with its expertise in single-cell and spatial-omics analysis, the open microscopy environment with its expertise with dealing with very large images and representing data with standard formats like the OM and GFF, and then a PARI team for interactive visualization and annotation. So together, we decided to build a framework that could address these challenges, and we call it the Spatial Data Framework. And here I want to emphasize that the goals of the framework is to provide an infrastructure for storing, manipulating, and visualizing data, and that we rely heavily on the Python geographical information system libraries. What is a known goal is that this is not an analysis library. We are not building the new SquidPy. We are building a tool that the SquidPy can rely on to make it more general for different type of data. And let's see how the framework is structured. We start with an abstraction. We represent the different type of spatial elements, the various geometries in memory, and we represent both raster types like images and segmentation masks, and vector type like point, circles, and polygons. These geometries can be annotated with feature tables, and on disk we write and read back using the OMNZFS specification, and we contributed, and we are still in the discussion of improving the specification when there was the need for this. The storage is both on disk and on cloud, and the spatial elements can be combined in any order, any number, with different spatial alignments, which are supported by coordinate transformations between 2 or more coordinate systems, and everything. This metadata is still compliant to the OMNZFS specification. And this allows, for instance, to look at the data in space, like in the physical space, in micrometers, but you could also be interested to look at the data in, for instance, the pixel space, which is what the technologies give you as output for the images. We also implemented part of a framework to interface with standard technologies. So we have ways to read the common formats like Cinium, Visium, Cosmex, and so on. And this can be used then to save and read to ZAR and NZFF. This, on top of this, also the memory representation can be used to visualize the data both statically using Matplotlib derived visualization tools or interactively with Napari. And together, this means that we can start from any technology, have an in-memory representation that is common for all of them, save it in a standard format with a community agreed specification, and then visualize it interactively. And these are some examples of the Napari views from various types of technologies with this unified interface for geometries and annotations. I want to now dive deep in an example that we describe in a recent preprint about the framework. And this is an example on the breast cancer data set with Visium and Cinium data that I briefly introduced before. So the data has come from 10x Genomics, is not aligned. Here there are the 2 images from Visium and Cinium, and this can be aligned using Napari spatial data by setting some landmark points and then deriving and saving back to disk the coordinate transformation to map the data in the same coordinate system. And here is an example in which with an iPad we can save some annotations in the tissue. You can see that we can zoom in and pan efficiently thanks to the ZAR multiscale and Sankt representation, and we can annotate regions of interest that can then be used downstream programmatically in our computational pipeline. And to perform this type of processing of the data, we implemented in the framework also some operations. So it's not just about visualization. So one of these is spatial queries that allows to crop with rectangles or polygons the data from any layers. We use this to be able to tile the data set. For instance, you can tile the data set around each cell by cropping out images, geometries, and annotations. And we show an example in which you can use this to interface with deep learning pipelines. And finally, we implement one extra thing that we implement is the feature aggregation that allows you to combine signals from different layers using any type of geometry. For this, I want to show you an example in which we took the region of interest that we annotated with the iPad, and this denotes some immune infiltration compartment of the tissue, and we wanted to quantify fractions of cell types from the Cinium modality of the data within this region. And this is an example of the cell type fractions that are computed. I also want to talk about interoperability. So with spatial data, what we could achieve is that we can connect different components of the Python world. So we rely for the memory representation on geospatial libraries, and we interface with the SCVerse tools for a single cell data analysis. There is an interface thanks to the tiling operation to PyTorch and with Napari. But also, if we consider the on-disk representation, we now create a link to different programming languages, and we are collaborating with R developers and JavaScript developers to make it possible. In particular, Vitessce will be available to read the data in NZFF. And finally, I want to also spend some time talking about the limitations and the current work that is being done on the framework. So there is, we learn a lot from the feedback from the users, and we are currently improving the ergonomics, the performance, robustness, and also adding some wish lists that the users added. And here I would like to to spend more time to talk with you about limitations, but I only have 10 minutes, so I will just focus on one thing that is the most important probably part that we want to improve is supporting multiple tables, which is a limitation now of the object that we had for a developmental reason, and the users ask for this. And I will be working with Lotte and Benjamin the next month on this. So to conclude, I talk about the spatial data framework, which is composed of several Python packages for in-memory representation, reading from disk, and visualizing interactively and statically the data. And we rely on the OM and ZFF specification to save and read the format. And here I want to thank all the collaborators that made this possible, in particular Giovanni, Isaac, and Kevin, which are co-first author and core developers of the library. I want to thank our PIs, Oliver Stegle, Fabian Theis, Wolfgang Huber, and Moritz Gerstung for their support, and the San Zuckerberg Initiative for the funding. Thank you for your attention.
That was great. First question from online. What is the computational scalability of this format? And as people are working with larger and larger data sets, how well does this handle it? What type of infrastructure might you need?
Yeah, so the key component on this is the fact that we rely on geographical information system libraries for Python, which are in particular X-ray, Dask, and GeoPandas. And from the raster part, this is already scales up to terabytes, because this combined with the NZFF, which provides a multi-scale representation and chunk representation, gives you basically an algorithm that is like Google Maps. If you zoom in, you load a higher resolution of the data, but for a smaller portion, so it's very performant. And together with Dask, this gives lazy representation and lazy computation. For the vector part, we rely on GeoPandas, which is built on top of PyGeos and other geospatial components. And we could GeoPandas supports spatial indexing, so some operation like querying and aggregation could use this. We are not using this yet, but we have a developer that will start working on this soon. So right now, I will say it scales like for hundreds of millions of points. It's like possible, but there are data sets that are bigger, also like billions, and this will be possible with this indexing.
Awesome. And then in terms of analysis pipelines, what's needed for developers in order to get this to work for their pipeline, and how do you envision deciding which ones are going to be directly integrated, which ones will just be interoperable with in terms of like downstream?
Yeah. So I answer this in two ways. One is to get started, and then what is required. So to get started, we have multiple documentations and example notebooks that show how to use the current framework within the Python ecosystem. And the second part is that regarding interoperability, we are currently working on making it easier, and we have a hackathon, for instance, next month to work on the R part. So we'll create some notebooks that show how to perform this. Right now it's possible, but it's a bit more rough. There is a paper from this year on OMSR in which it shows interoperability between different programming languages for the raster part, but for the vector part, we will have to do some extra work.
Awesome. Well, join me in thanking Marco for a really great talk.
Thank you.
And with that, we will take our break. We will come back here in 23 minutes at 10:40.